Hello students, today we'll be talking about neuropsychology, neural mechanisms of learning. After studying this module, you shall be able to learn about chemical changes that accompany learning, learn how learning may be defined in a neurological paradigm, learn neural mechanisms of classical conditioning, learn neural mechanisms of operant conditioning, learn the neural mechanisms of perceptual learning, understand the concept and neurological basis of synaptic plasticity in learning. Introduction. The brain constantly rewires itself to become an organ that is physically different from what it was in the preceding moment. Never resting, it churns through new experiences daily, incorporating some new information into its networks of stored information and discarding others. The ever-changing nature of human brain gives rise to two questions that have been bothering thinkers for centuries. They are: do psychological processes end into biological processes? Can we physically locate all our experiences? Aristotle's teacher, Plato, reasoned that the mind had to exist inside the brain because of geometry and pure logic. The brain was round, he said, and close to the perfect roundness of the sphere. It also inhabited the part of human body closest to heaven. Plato and other Greek philosophers theorized about the existence of a force that keep people alive and left them at the time of death. They called this force psyche or soul, and several authors said it resided in the brain. Centuries later, Galen, a Roman physician who lived in the eastern Mediterranean in the 2nd century of the Christian era, went beyond such mental exercises to test the brain himself. He took a more hands-on approach and cut the sensory and motor fibers in pigs' brains to observe results. Galen became the first person to speculate that certain functions are carried out by specific parts in the brain. He made rudimentary descriptions of the body's major organs and fleshed out description of what he saw as the varieties of human spirit. Thus was born the conundrum that has sparked debate for many centuries. It is called the mind-body problem. Over the past several decades, there has been a gradual merger of two originally separate fields of science. They are neurobiology, that is, the science of brain, and cognitive psychology, the science of the mind. Recently, the pace of unification has quickened, with the result that a new intellectual framework has emerged for examining perception, language, memory, and conscious awareness. This new framework is based on the ability to study the biological substrates of these mental functions. A particularly fascinating example can be seen in the study of learning. Elementary aspects or the neuronal mechanisms important for several different types of learning can now be studied on the cellular and even on the molecular level. The analysis of learning may, therefore, provide the first insights into the molecular mechanisms underlying a mental process and so begin to build a bridge between cognitive psychology and molecular biology. Definition of learning in terms of neurology. Learning and memory make each individual unique. Even before birth, the human brain takes in sensations, processes them, and begins to encode them into trillions of synapses. Those connections and the electrochemical firing patterns that unite them make the brain an organ different from what it was a moment before. As new connections form in response to stimuli and then become strong through repeated use, the brain integrates new information and stores it until needed. Without learning, memory would be a little more than a clockwork. Learning and memory work together. Learning produces changes in the way we perceive, act, think, and feel. It does so by producing changes in the nervous system in the circuits responsible for perception, as well as those responsible for the control of motor activities. From a purely psychological point of view, learning may be defined as the relatively permanent change in behavior acquired through practice and experience. Neural definition of learning, however, may be summarized as structural and chemical alterations that are acquired in the brain on encountering an external stimulus. Neural mechanisms of classical conditioning. When Pavlov first described the process of classical conditioning, it was fairly behavioral in nature, based on observable facts of the experiments. Later, as the belief of the role of neural processes was understood, the neuropsychologists and cognitivists began to explain it in terms of the neurological mechanisms involved. Neuroscientists have studied the anatomy and physiology of classical conditioning using many models, such as the gill withdrawal reflex in aphasia. In classical conditioning, the stimulus, or S, triggers the response, or R, of an organism. With the exposure of the organism to the stimulus, reflex results. Reflex is the involuntary behavior which comes from within. One of the most common reflexes seen is the blinking of the eye whenever air is blown into it or a foreign object approaches it. The concept of reflex with no conscious control is incorporated in classical conditioning. Pavlov developed a model for classical conditioning. It involves the following. First, the unconditioned stimulus, or the US, triggers the unconditioned response, or the UR. This means that without learning, a stimulus can elicit a reflex. Reflex is called the unconditioned response because it is involuntary and we do not need to learn it for the event to occur. Next is the neural stimulus, or the NS. It is a stimulus that does not elicit the unconditioned response. This means that when this stimulus is presented to the organism, it would not execute the same response he showed when it encountered the US. US and NS are repeatedly paired and presented to the organism in conjunction. The pairing of the US and NS transforms NS into a conditioned stimulus, that is CS. This means that whenever the previously neutral stimulus is presented alone, that is US is not showed anymore to the organism, it causes UR to occur. But this time UR is changed into conditioned response, or CR, because the response is elicited due to conditioning. Therefore, CS elicits CR. Pavlov believed that classical conditioning reflected a strengthened connection between a brain area that represents CS activity and a brain area that represents UCS activity. Because of that strengthened connection, any excitation of the CS center flows to the UCS center, evoking the unconditioned response. The connection between these two areas was labeled as the engram. Lashley in 1950 studied such engrams by severing the connection between several brain areas after training subjects in classical conditioning. No specific place where such engrams are made was discovered. Learning was found to take place in diffused areas. A model of the neurological basis for classical conditioning was developed by Weinberger and his colleagues. These researchers used the conditioned emotional response. Some neutral or conditioned stimuli, in this case a tone of a given frequency, is associated with an unconditioned stimulus, in this case food, which eventually comes to elicit a conditioned response, in this case an autonomic arousal. The autonomic arousal also accompanies secretion of epinephrine, an excitatory neurotransmitter, as well as a hormone of the adrenal medulla. High level of epinephrine raises the level of blood glucose. High glucose level facilitates the process of learning and memory. Role of amygdala. At the level of neurological structures, the response to food travels via a sensory pathway that runs through the central nucleus of the amygdala to the somatosensory cortex. This is not surprising given that the central nucleus plays an important role in emotion. The auditory stimulus, the tone of a given frequency, travels via a sensory pathway that passes through the medial geniculate nucleus of the thalamus and on the auditory cortex. Pathways of the two are diagrammatically represented. The amygdala plays an important role in organizing a pattern of emotional responses that are provoked by a stimulus US. Central nucleus of amygdala is activated. The information about conditioned stimulus reaches to the lateral nucleus of amygdala. This nucleus also receives information about unconditioned stimulus from the somatosensory system. These two sources of information converge in the lateral nucleus, resulting in learning the conditioned response. Neurological association takes place, especially in the nucleus basalis, a brain structure rich in acetylcholine. Neurons which sends projections diffusely into the cerebral cortex. When the neural signal associated with the root shock reaches the central nucleus, an auxiliary pathway which goes to the nucleus basalis is also activated and results in a general diffuse activation of the cortex. Most importantly, for our purposes, these results in an increased level of sensitivity in the auditory cortex, so that it is now more sensitive to neural input from the medial geniculate nucleus. In this manner, via the nucleus basalis, acetylcholinergic neurons and the auditory cortex, the unconditioned stimulus, that is the food, and conditioned stimulus, that is the tone of a given frequency, come to be associated. Operant conditioning. Instrumental or operant conditioning is the means by which we profit from experience. If in a particular situation we make a response that had favorable outcomes, we tend to make the response again. Role of basal ganglia. Operant conditioning entails the strengthening of connections between neural circuits that detect a particular stimulus and neural circuits that produce particular response. Therefore, instrumental conditioning involves various regions of sensory association cortex where the perception takes place and the motor association cortex of the frontal lobe. There are two major pathways between the sensory association cortex and the motor association cortex. They are: first is direct transcortical connections, and the second, connections via basal ganglia and thalamus. Both of these pathways seem to have a crucial role in operant conditioning. However, they function differently. The direct transcortical connections are involved in the acquisition of episodic memory and in acquisition of complex behaviors such as following instructions. It helps in establishing rules that leads to that, and thus in future, in case of similar situation that presents similar stimuli, these connections offer information as learned earlier. Evidence suggests that as learned behaviors become routine or automated, they are transferred to basal ganglia. The basal ganglia receive information about the stimuli that are present and the responses that we are making. When these are repeated, it is stored here. And once again, the transcortical connections are free for other tasks. The outputs of the basal ganglia are sent to the frontal cortex, to the premotor and supplementary motor cortex, where the plans of movements are made. Finally, it travels to primary motor cortex, where the plans of actions are executed. Perceptual learning. Learning enables us to adapt to our environment and respond to the changes that occur in it. It provides us with the ability to perform appropriate behavior according to the situation. Learning to perceive is highly neurological in nature. Perceptual learning is a lifelong process. We begin by encoding information about the basic structure of the natural world and continue to assimilate information about specific patterns with which we become familiar. The information for this kind of memory is largely represented in the temporal lobe. This idea originated from the finding that lesions of certain portions of the temporal lobe lead to the loss of ability to recognize faces, a syndrome known as prosopagnosia. Role of visual cortex. When the unconditioned stimulus is visual in nature, the visual cortex is actively involved. Objects are recognized visually by circuits of neurons in the visual association cortex. The visual cortex receives information from the lateral geniculate nucleus of the thalamus. Within the primary visual cortex, the individual modules of neurons analyze the information from restricted regions of visual scene. The information is collected in the subregions of extrastriate which surround the primary visual cortex. The ventral stream of the visual cortex is associated with the object recognition. The dorsal stream is involved in the perception of location. Learning and synaptic plasticity. Role of hippocampus. Learning must involve plasticity. Plasticity denotes changes in the structure and/or biochemistry of synapses that alter their effects on postsynaptic neurons. When relearning of some structures is involved, hippocampus takes up a big role. Synapses have plasticity and small alterations in the learning structures go on happening. Long-term potentiation is essential for the consolidation of learning. Long-term potentiation, that is LTP, denotes a long-term increase in the excitability of a neuron caused by repeated high-frequency activity of that input. The study of long-term potentiation in the hippocampal formation has suggested that such mechanisms might be responsible for at least some of the synaptic changes that occur during learning. High-frequency stimulation of axons in the hippocampal formation strengthens synapses. It leads to an increase in the size of excitatory postsynaptic potentials in the dendritic spines of the postsynaptic neurons. Associative long-term potentiation can also occur in which weak synapses are strengthened by the action of strong ones. In fact, the only requirement for long-term potentiation is that the postsynaptic membrane be depolarized at the same time the synapse is active. Hippocampal formation is a forebrain structure of the temporal lobe, constituting an important part of the limbic system and includes dentate gyrus and subiculum. CA1 and CA3 are important structures of the hippocampal formation. In the field CA1 and dentate gyrus, NMDA receptors play a special role in long-term potentiation. NMDA receptors are specialized ionotrophic glutamate receptors that control the calcium channels. These receptors are sensitive to glutamate, control calcium channels, but can open them only when membrane is depolarized. Thus, the combination of depolarization and activation of NMDA receptor causes the entry of calcium ions into the cells. The increase of calcium activates several calcium-based enzymes, causing insertion of AMPA, another receptor. AMPA receptor is an ionotropic glutamate receptor that controls the sodium channels and produces excitatory postsynaptic potentials. These changes result in structural alterations in the shape of dendritic spine. Long-term potentiation requires protein synthesis, which appears to take place in the dendrite adjacent to dendritic spine. Long-term depression occurs when a synapse is activated at the same time that the postsynaptic membrane is hyperpolarized. The process of long-term potentiation may be summarized below. After repetitive stimulation of a neural pathway, a synapse will show two signs of increased efficiency. A single stimulation causing a more rapid increase in the excitatory postsynaptic potential, that is EPSP, and a single stimulation recruiting more cells that reach the action potential threshold. Chemical changes during learning. Synaptic activity in hippocampus releases glutamate, that is excitatory, and calcium ions. NMDA receptors is blocked by magnesium ions and AMPA receptor opens. AMPA allows in sodium ions, excitatory postsynaptic potential, that is EPSP results. Activation of postsynaptic dendrite releases magnesium ions from NMDA receptors. Glutamate binds with NMDA receptor. NMDA pumps calcium ions into cell and causes action potential faster than sodium ions. Calcium ions used to synthesize neurotrophins. Neurotrophins affect synapse, more, larger, stronger connections. Long-term potentiation causes AMPA receptors to move into the dendritic spines and appears to cause the formation of new synapses. Chemical reactions, not yet fully understood, also cause the release of glutamate. Long-term potentiation also requires synthesis of new proteins, including components of cytoskeleton, protein kinases, and receptors. It is also accomplished here in this process. Summary. Learning accompanies structural and chemical alterations in the brain on encountering an external stimulus. The central and lateral nucleus of amygdala plays significant role in the process of classical conditioning. Direct transcortical connections and those via basal ganglia and thalamus are involved in operant conditioning. Visual cortex plays important role in perceptual learning. Long-term potentiation of consolidation of learning takes place in the hippocampal formation, where NMDA and AMPA receptors facilitate the process via their chemical mediation. Thank you.